Chapter 15 is all about the idea of chromosomes. Okay, and to review, the basic idea is this. This is a chromosome. Chromosomes are made of DNA. Talk more about DNA in the next chapter. Chromosomes are made of DNA. On each chromosome are specific genes. Genes are at specific loci or a locus or a location on every chromosome. So if in humans we have 46 chromosomes but in 23 pairs, that means there's 23 different chromosomes. We have approximately, in the last few years we figured out, we have somewhere between 20 and 30,000 genes. So if we have between 20 and 30,000 genes, that means that in our 23 chromosomes that can contain genes, because remember, they're paired, so that means that the other one of the pair has the same genes, maybe a different allele for the gene, but the same gene at the same locus. So if we have 20 to 30,000 genes, that means we have many genes per chromosome. That's the first and most important idea from this chapter. We have many genes per chromosome. So if we review meiosis then a second, and we talk about that if we look at chromosome and inheritance, this diagram, 15.2 in your book, is an important one. Because it shows, now this shows only one gene on each chromosome. Okay, one gene on each chromosome. So the pea seeds undergo meiosis, or a sperm cell undergoes meiosis, or an egg cell, and it passes on only one copy of each gene. And so here we have this cross this. We're going to get all the kids will be this. Why? Because the genes are carried on their specific chromosomes. You get a chromosome from mom with the yellow allele, or the round or wrinkled allele, and you get a chromosome from dad with a round or yellow allele. Round or yellow or green allele. Okay, and vice versa. So now, in each of these parents, if you were to cross them, or they each do meiosis, now comes Mendel's law of segregation, that you're only going to pass on one copy of each gene to each offspring. Okay, and the reason you do that is you're only going to pass on one of these two chromosomes to your offspring. You aren't going to pass on two, okay, if this is chromosome pair 1 and this is chromosome pair 2, chromosome pair 1 contains the little r's and chromosome pair 2 contains the y's. You're only going to pass on one of each. Notice down here, haploid cells. So you pass on chromosomes with genes on them. The law of independent assortment says that whatever you're going to give is a random chance. You could give a little r and a little y, a little r and a big y. Or in this case, it's not so random. A little r and a big y, big R and a little y, big R and a little y. Or a big R and a big y, and a little r and a little y. And then fertilization would lead to our this cross this. Dihybrid cross 9:3:3:1 ratio. Then one day in the 1900s, along came a guy named Thomas Morgan, and he was looking at fruit flies. And he noticed something. He was doing some crosses with red-eyed flies and the mutant white-eyed fly. Mutant would be recessive in this case. Not always, but in this case it was white. And by the way, if you're looking at this in your textbook, when they do fruit fly genetics, for some reason, they use the term wild type as the dominant characteristic, and they'll show it with a plus. I'm going to use letters to represent the crosses. Okay, he was doing this. He was messing around with fruit flies, and he found out something really weird. That when he crossed red-eyed flies and white-eyed flies, if he crossed purebred red and purebred white, if he did that, sometimes he expected to get all that, right? All red. But sometimes he would get some whites. And they were always males. And that was a surprise to him. How come sometimes I get white males? Well, through some more research and a little bit of intelligence on his part, a lot of intelligence, the theory he came up with was this, and it happens to be true, that the gene for eye color in fruit flies, and you can look this up in the book and look at fruit flies, and we're going to do some more detail with this, is on the X chromosome. What does that mean? Well, that means that if it's on the X chromosome, females are XX and males are XY. That means that males only get one copy. Males only get one copy of that gene in any cross. So what that means is we say that if that's where the gene is, it is called an X or sex-linked gene. An X or a sex-linked gene or X-linked gene. Because females, I'm going to draw it like this, have two copies of it if they are homozygous dominant or purebred. A male will only get one copy of it. And I'm showing it drawn on the chromosome. If we did this cross, this is purebred red cross purebred red, and you can see they'll all be red. But what if we crossed some purebred red, if we had a heterozygous red crossed with a red male? Well, just by doing rules of probability here, what's this one going to pass on to its offspring? Well, remember that if you are a boy, you got this chromosome from your dad. There's a 50% chance of that. So it has a 50% chance of making boys. The female has a 50% chance of passing on this chromosome or that one. And so half the time you'll get a boy, and the half of the boys will be that. And that means half the boys will be the white-eyed phenotype. Okay? That's called sex-linked or X-linked genes. This diagram, 1510 in your textbook, shows different possibilities for gene linkage. Okay? You can cross a homozygous dominant female with a recessive male. And here's the Punnett square that results. Okay? 50% chance of getting boys, but all the kids will be dominant. If you take a heterozygous girl and cross her with a dominant male, 50% chance of getting a boy, 100% chance of having a dominant girl, 25% chance of having a recessive boy. You could cross heterozygous female with a recessive male. Carrier female, dominant male, recessive female, recessive male. And the last, okay, so those are some possibilities for passing on, and those are examples of X-linked or sex-linked crosses. Now, what this all means is, then, that if you have a chromosome, and on it is the gene for, let's say, flower color or eye color, and let's say you're brown eyes, so I'm going to draw 2 chromosomes here for you, and let's say you're heterozygous. And on the same chromosome, you have the gene, let's say, for being able to roll your tongue or not, and you are heterozygous for tongue rolling. But it just so happens that from your dad, you got the 2 dominants, and from your mom, you got the 2 recessives. When you pass, remember, you pass on chromosomes. So that means when you pass on, you're going to pass on one of these 2 chromosomes to your kid. Just one. Okay, if you're a male, it's going to be in your sperm. It's my lovely sperm tail. So that means that this parent, if this is true, if they pass on this whole chromosome, they're going to pass on both of those genes together because those genes are linked on the same chromosome. They're linked on the same chromosome. You need to understand that concept before we go on. Okay? You have 2 genes or more on the same chromosome. They're called linked genes. Okay, so let's talk about that a minute. So normally then, if you have linked genes, and let's say you have a heterozygous person like the one I just showed, okay, that they have the big B and the big R together, and the little b and the little r together, okay, and you cross them, we call this a test cross. You test cross with that. What would you expect to get? Think about that question for a second. What would you expect? Well, you would expect to get, because these go together, you would expect to get this parent passing on a big B and a big R together 50% of the time. This parent is always going to pass on a little b and a little r. So 50% of the time, you're going to have that. And the kid is going to be dominant. Is going to look brown eyes, roller. And the other 50% of the time, you're going to pass on that chromosome, that chromosome. So 50% of the time, you're going to get a blue non-roller. By the way, all of these genes have been made up. Okay, so if you understand that concept, imagine Thomas Morgan's surprise then when he did something like this. He knew the genes were linked, and yet when he did something like this, he got numbers, say, like this, where the brown rollers, the double dominant, he got, instead of 50%, he got something like 42%. And the double recessive, blue non-roller, he got something like 42%. And the combination, so that would be something like brown non-roller or blue roller Okay, so one of them is, so basically you're getting this or this. Oh, I didn't do the math right. That adds up to too much. We have 84%. We got 8% of those. How did those show up? How does that happen? Well, what this is actually showing, what that is actually showing is this. Remember, when you make chromosomes, so here you are with your big B and your big R, and the one from your mom, the little b and the little r. When you do meiosis to make copies of these chromosomes pair up. And remember that sometimes when they pair up, they cross over. Oh, shoot. They cross over and exchange parts. Cross over and exchange parts. And so, if you do this, and 16% of your offspring are the mix. That means 16% of the time when these two things crossed over, it crossed over between those two genes. And we say then that those two genes are 16 map units apart on a chromosome because they cross over between them 16% of the time. If the two chromosomes were, if the two genes were closer together, and then they cross over, you would expect the chances that that happens between these two things to be less. So that would make that number less. So you're doing a problem with fruit flies. And the wild type is red eye, let's not use eye color, is black body, normal wings. They're on the same chromosome. They're linked. And you have gray body and vestigial wings, and we'll talk about that later. It's little crumpled wings. This is double recessive. So this would be like little b, little b, little n, little n. They're linked. They're linked, so they're going to be passed on together. But you do this, and you get, I don't know, 300 black normals. You get 300 gray vestigials. And in the middle, you get, to make the math easy, I'm trying to think of how to make the math easy, 40 of either black, vestigial, or gray normal. We call these, in linked gene talk, recombinants. They are the result of crossing over. If we want to know how far apart the genes are, we total up the total number of organisms. 300 plus 40 plus 300 is 640. And we want to know on the chromosome how far apart are these genes? How far apart are the black normal gene? Well, we take the frequency, 40 out of 640. If I do that math like this, I get 1 over 16, which is about, I'm trying to do this math in my head and I'm not very good at it. Whatever that is a fraction. That approximately 6 map units apart. So then, if on a chromosome you have 3 genes, and you know, and you don't really know where they are, but you know that, I'm just going to say they're genes A, B, and C. And you know that A to B is 10 map units, and A to C is 15 map units, and B to C is 5 map units. You can figure out where these are, right? Because that would mean that B to C is 5 map units, B to A is 10 map units, and A to C is 15 map units. And now we have mapped the chromosome. And especially if we can figure out where to start. That's the first part, and probably the most important part, well, the most important part for problem solving of chapter 15.